Hello, welcome to the lab. My name is Ellie and I'm a graduate student who studies developmental biology. Specifically, I make movies of the fruit fly embryo as it develops. Let me show you around. This is my lab bench. It is where I perform most of my molecular biology. I work with DNA that is in various types of solutions. I use pipettes like these, as well as tubes like these to hold the DNA. I perform reactions that amplify the DNA, and then ligations that combine different strands of DNA together. This is how I create transgenes that encode for fluorescent proteins. I then inject this DNA into fly embryos to create a new strain of flies. Now, I'll show you the fly room. This is what we call the fly room. It contains many bottles of different types of flies, as well as hundreds of vials of different fly strains. The tape on the bottles and vials tells you the genotype of the flies inside. Sometimes, not all flies in a bottle will have the transgene that we introduced. So, we have to look at genetic markers that tell us whether a piece of DNA is present. These can be curly wings, white eyes, or dark body. Wild-type flies have straight wings, red eyes, and a lighter body. My favorite genetic marker is fluorescent eyes. Let me show you what they look like. To look at the flies under the microscope, we first have to make them unconscious so they don't fly away. This plastic pad has small pores in it that emit CO2. The CO2 then displaces oxygen in the air and causes the flies to go to sleep. But don't worry, they'll wake back up again. A lot of my research involves making movies of developing fly embryos using a confocal microscope. To get embryos that express fluorescent proteins, I take male and female flies that have the correct transgenes and then put them in a plastic cage together. The flies then lay embryos on this bottom plate here. I then pick up the embryos with tweezers and place them in this small plastic bucket. I then pour bleach on, which removes the outer membrane. I then transfer the embryos onto a plastic microscope slide such as this. Now, the embryo is ready for imaging. I'll show you the microscope. Here is the confocal microscope. It uses lasers of different wavelengths to excite fluorescent proteins. The proteins then emit light, which is then detected by the microscope and converted into an image. To set up the microscope for imaging, I first place a drop of oil onto the microscope slide, and then a drop of oil onto the objective of the microscope. This oil prevents refraction of light and allows for more light to be picked up by the detectors of the microscope, creating a sharper image. I then place the microscope slide very carefully onto the microscope. Here's the computer that controls the microscope. Using these bars, I can set the color of the laser that I use to scan the embryo. Down here, I can set the wavelengths of light that the detector picks up. The computer then converts the detected light into an image.
The development of a fly consists of a few main stages. First, female flies lay embryos. The embryos then develop and hatch to form larvae. Larvae then grow and eventually form a pupa. Finally, fully formed flies hatch from the pupa. My research focuses on the early embryonic development of the fly, so this stage here. The embryo starts off as a single giant cell with a single nucleus. The nucleus then divides until the embryo is filled with hundreds of nuclei. The nuclei then migrate to the surface of the embryo. Then, after about 2 hours into development, individual cells form around each of the nuclei. Now, let me show you some videos of embryonic development. This movie was created by the Confocal Microscope by collecting light transmitted through the embryo. You can see that around the outside of the embryo, there are little circles, which are the nuclei. Basically, what you're looking at is a slice through the embryo, with the dark being the middle here, and then all the nuclei around the outside. Each of the shutters that you see corresponds to a nuclear division. So when the one nuclei becomes two, and so forth. You can see that after the 13th nuclear division, the cleavage furrow proceeds through from the outside of the embryo to the interior and encases each of the nuclei in cell membrane. So this is the dark line right here that's moving inward. Once the cells are encapsulated by cell membrane, the process of gastrulation begins. So right now, this is the process of cellularization. And now when the cells all move inward and there's this swirling process, that is the process of gastrulation. This is a fluorescence movie that was created by the confocal microscope. It is a zoomed-in portion of the surface of the embryo. The blue you see is fluorescence from fluorescent proteins inside the nuclei. This movie shows the process of nuclear division. This is another fluorescence movie created by the confocal microscope. The green spots that you see are spots where the information in DNA is being read. The spots show the location where molecules known as messenger RNA or mRNA are being produced. Here's another fluorescence movie created by the confocal microscope. All the little spots that you see are nuclei. Different nuclei contain different types of proteins. The stripes that you see that form across the embryo is a specific type of protein that serves to pattern the embryo.
Thank you for joining me in lab today. I hope you have enjoyed learning about my research.